Why should I learn about microbes for fresh cut processing? There are many aspects of quality and safety that involve the microbiology associated with products in the field, whether it's decay or spoilage organism, but particularly for food safety. So understanding human pathogens and their biology is really important, their dynamics and survival and dispersal on the field, whatever is on the product in relation to your goals and objectives, in washing and disinfection to prevent cross-contamination, and I'll emphasize that in my next talk, and different interaction with leaf microbiota. There are new advanced technologies evaluating what's on the product. So I'm not going to cover much in detail that, but we're still trying to understand the interaction microbes plan and how they can benefit or help and protect us later. All right. So the microbial world is very diverse, but just to keep in mind, less than 10% of the organisms that we currently recognize and identify can be cultured or grown and actually be visualized and studied. So most of the stuff that you see on plate is not the total of the microbes that are out there that can be grown. There are modern techniques looking, like I was saying in the previous slide, at the microbiomes, another way to study what we cannot culture, but the thing is that we probably haven't figured out yet how to culture them, how to grow them, so we can study them. So that covers a variety of different things, from bacteria to fungi to viruses and to parasites. And this is a question that we get asked a lot, like how big is a microbe? We're talking about a microorganism that are microscopic. So in relation to a human cell, the average human cell is like 25 to 30 micron. There on the left, you have Salmonella that is about 1.1, 1.5 micron in length. So if you convert that into inches, 1 micron is approximately 0.00004 inches, which it will be in our, from our perspective, like one step for a walk across the US. So we're talking about very small small microbes. Also like hair, the thickness of hair on average is about 75 microns. So you can fit 75 Salmonella alongside in the diameter of a hair. Sources of microorganisms in food. You have to be aware that microbes of concern in food safety that can impact quality and safety can come from any number of different sources and can reside pretty much anywhere. So I'm not going to go through all these. I know some of the stuff for you is, for you guys that are involved in food safety is maybe like very basics, but for those of you who are not very familiar, pretty much microbes are found pretty much everywhere in your hands, in water, and packing material on the surface and raw ingredients. And I'll go into more details later on. Raw foods contain viable living microorganisms. We can't see them, just the challenge. But most of the time, they don't change the appearance or the flavor or the odor of the product until it's too late. For example, here in these tomatoes on the right, one of them is contaminated with 10 million cells of Salmonella. Another one is contaminated with 10,000 spores of Alternaria. So can you guys guess which one is which one? Which one would you eat? Which one would you throw away? Like no way to tell. Bacterial growth, on average, at room temperature under ideal conditions, the doubling time for these microorganisms is like 30 minutes. So if you start out with one cell, which is very rare to have one cell alone, but let's say for the sake of this representation, you start with one cell, in half an hour you have two, in an hour you have four, so each cell, each bacteria will duplicate, and each one then in turn will duplicate again. So that continuously, this is under ideal conditions. So in about 10 hours, which are the incubation time for most of the rapid tests, you have millions of cells. So why don't bacteria take over the world? Maybe they already have and we don't know it. Traditional enrichments that tend to go longer, like FDA methods, they will go at least 18 to 24 hours. Then we're talking about in the billions. Factors that influence microbial survival and growth. Well, you have obviously food. They need to eat something to grow. Acidity, or the pH, temperature, time, oxygen, and moisture. And I'm going to go through all of them. A good way to remember, it's probably not a very politically correct acronym. It's FAT-TOM. So anyway, so this is like the way we were taught to help you guys remember it. So let's go through FAT-TOM. Well, let's just start with the pH. The limit of pH for growth of some key microorganisms. There's certain pH range that these bacteria can grow or survive. For example, E. coli 157H7, the really serious human pathogen, can grow in a range of pH from 4 to 9. So from very acidic to very alkaline. And then temperature, also a broad range at which they can grow or survive slowly. For example, again, the EHEC group, the enterohemorrhagic E. coli, like E. coli 157H7 and Salmonella, their ideal or optimal growth is around our body temperature at 98 Fahrenheit, 37 Celsius. So we call these mesophilic organisms. But then you have those that can grow really cold temperatures, like Listeria, and some decay-related organisms like Pseudomonas or lactic acid bacteria. Cold temperatures around 41 Fahrenheit or 5C, like Erwin was saying yesterday, it's not really a control point. It will take a few cells, some of them will die, but most of them will still survive and they will be there. So lowering the temperature is not a way to control your microbial counts. Oxygen availability can limit growth of bacteria, Pseudomonas, and like I said before, it causes decay and spoilage. At refrigerated temperatures, needs high levels of oxygen. Then you have anaerobic bacteria that can grow with no oxygen, aerobic bacteria that requires oxygen, like Pseudomonas. And then, like Jeff was saying yesterday with modified atmosphere packaging, oxygen levels are reduced, but they're not zero. They still have some oxygen. So whatever is still on the product will still grow inside very slowly. It will survive, but still grow. And that's the case for E. coli and Salmonella. Bacteria frequently found internally in vegetables before cutting. Even in properly grown fruits and vegetables, bacteria are frequently found internally. Many of them, they will colonize a variety of different fruits. Most of them are neutral or beneficial in terms of competitive exclusion of human pathogens, but they can become involved in senescence, trigger spoilage. So as a natural physiological spoilage is going on, that activates them to start growing, and then they themselves accelerate senescence, spoilage, and decay. Some are opportunistic human pathogens, and then you have some that are both human and plant decay pathogens, and we call them cross-domain pathogens. Raw foods contains microorganisms. We already established that. Suboptimal temperature is the major challenge in fresh cut, even at very good refrigeration temperatures. There's a variety of plant pathogen or spoilage organisms that can get involved in decay, like in this case, Pseudomonas soft rot decay in celery, and leuconostoc decay on carrots on the top. Now I want to clarify a few things. There are no established and validated microbial limits for produce. there is insufficient evidence that aerobic plate count or heterotrophic plate counts of the total bacteria could serve as an effective quality indicator or indicator of food safety risk. Some of that, some of the new techniques, the evolving techniques with rapid DNA or RNA-based genetic tests are helping that, and they're getting more affordable, but as of just looking at the total plate count, the total bacteria count, let's say on the finished product, it's not going to tell you anything. If you're not doing something before, during the, let's say, the washing and then after, then that number at the end, it won't say much. I'll explain or go more in detail with that in my second talk later today. So now I'm going to, in the second half of the presentation, and I'm going to try to cover all the slides for pathogen or actually microbial testing. So pathogen testing programs, why do you test? Well, there may be regulatory recommendations. It may be a requirement. It may be part of the testing program for a food group. It may be a requirement from the buyer. You may decide or you may feel the necessity to have pathogen testing based on your product history, so maybe you want to do that, like a volunteering approach to safety. Or you just do it because everybody else is doing it. It's seems like a good idea. Well, one thing I want to emphasize that after people doing hundreds of thousands of tests, at some point you will find positives in the pre-harvest, and the raw material, and the finished product. And these might help prevent this product from reaching the market. So I would argue that testing is within the commodity you work with, it would be a good idea. But then while there are no scientific validity to support that you're doing this in terms of addressing the probability of detection. And I'll go through the probability of detection in a couple slides after this. So it's a very poor correlation with shelf life. It's sometimes, not sometimes, most of the time, pathogen levels, unless it's a very heavy contamination, the pathogen level on the product is very low. So it's unlikely that you will detect them. So you're wasting your money and your resources for tests, knowing that the chances are that you're not going to find them. Then practical tests not available. That depends on what you are after, what's your target organisms. Or maybe another option is that you want to know. Microbial testing requires a very clear understanding of your goals, assumptions, and characteristics of the testing program. And then in writing, you want to have the understanding of the limitation and define also the limits of your program. Also in writing, you want to have the defined corrective actions or corrective measures. And also the data disposition policy of all these results, where they're going to go. Sample size, for non-pathogens, is typically 10 grams of the sample of the product. It could be an area. If you're doing environmental, under the environmental monitoring program, you might surface in an area looking for Listeria species. For pathogens, it's typically 24 grams. It should be higher. Our field research data shows that it's better that you do smaller samples. Like, let's say you have 1.5 kilos of lettuce and you want to test it. You could do 4 samples of 375, or you can do 60 samples of 25 grams and then composite them. So at the end, you have the same amount of samples testing. So you have more smaller samples that you composite, and then you test. But then it depends also on the product and what commodity you're working with and the organism that you're after. Organisms, what to test, there's a difference between index organism like aerobic plate count or total bacteria count, and looking for very specific spoilage microorganisms like Pseudomonas. To predict shelf life, that tends to be better than looking at just the total bacteria in the product. Indicator organisms, you have coliforms, thermotolerant coliforms, or fecal coliforms, E. coli, coliphage, and I think in the PDF online, I left out that I don't have here, kind of like something that Trevor reprinted on tolerant bacteria and how to divide them. So if you see the graph, it will help you visualize when we talk about all these different indicator organisms. Microbial testing and verification in the processing plant. A lot of the emphasis on environmental monitoring programs, it's on listeria species or listeria mono. And biofilm became like a catchphrase. So if you find listeria and you find it again, everything explained by just saying, oh, biofilm. They make biofilm, and that's why you can't get rid of them. We have collected several environmental isolates of listeria monocytogenes from different facilities, mainly raw agricultural commodities, not so much from fresh cut produce. And we characterized them, and we found that many resin isolates don't appear to have a strong biofilm production capacity. They're just really good at surviving where they are. They might be also opportunistic. There are other bacteria that can produce biofilm and then just happen to land where they are, so they can also be protected by biofilm from other bacteria. But it's not that every listeria monocytogenes found on a packing house will be a strong biofilm producer. Current verification of food safety programs. In process product, you tend to do, like I was saying before, total plate count, total coliforms, E. coli, EHEC, and E. coli 157H7, although 0157 is an EHEC. Anyway, that's for another time. And then Salmonella. For finished product, you do again also total plate count, E. coli, the generic E. coli, EHEC, the EHEC group, including 0157 or just 0157, Salmonella, and Listeria monocytogenes. Rapid quantitative assessment, just like ATP swabs, often used to evaluate cleanliness. There are rapid or semi-quantitative assessment procedures. They evaluate the cleaning and surface disinfection procedures, and that's about it. It's not as quick as the typical ATP. It takes some time, especially if you want to enumerate, you want to count, and if you're going to do presence absence. But it tells you nothing about the pathogens, okay? So you're doing just this total bacteria, pretty much. So you can work with the lab for third lab parties, but kind of accept the system that they pick. You can suggest a system, but yeah, go by what they pick. When selecting a system, the thing that you want to keep in mind is that it has to fit your application and your commodity. It has to be invalidated around how you are going to use it. And also make sure that the company provides very good technical support. And a commitment to the produce that you're working with. Collecting samples, we talk about sample size. Now let's go a little bit over how we're going to collect them. One of the most important things when it comes to sampling is what you define by a lot. So there are many different definitions, and depends on how you define it. The lot is going to dictate that if that sample from that lot is positive, then if you're going to destroy that product, all the product has to be destroyed. But if you call the lot as everything coming from that one field, and that's your lot, and then positives coming from that, and like in the sample, like 4 pallet combination, you can't just get rid of one, whatever came from that one pallet, you got to get rid of from everything. So make sure that you take your time to define the lot well. Well, then again, samples have to be selected randomly, and understand that the microbial sampling is a destructive process. And if done the traditional way, the results may be delayed for accuracy, but in the last decade, the test kit developers have done a really good job improving the specificity, the speed, and the accuracy of this test, and I'll go through that in a little bit. Sample may be dynamic or perishable, and can you sample and hold to ship? Well, that will depend on the commodity, how long you can hold your product. Key requirements of a pathogen test method. Exclusivity, I'm just gonna go and read through them. Exclusivity, effective in preventing positive reaction by non-target microbes. Inclusivity, effective in obtaining positive reaction by all forms of target microbes. Then selectivity, effective in obtaining positive reaction in presence of high population of non-target microbes. That's something that you will have in a typical enrichment. You're enriching everything. There are some selective enrichment, but still you are going to be enriching a lot of other things. And sensitivity, effective in obtaining positive reaction at lowest desired or required detection limit. And I'll go through the detection limit after in a few slides. So the pathogen detection timelines, or what it's usually called time to decision. The ideal rapid methods, like I was saying before, target these 8 to 12 hours. It may be longer for Listeria, probably about 20 hours. But you have to go through an enriching phase. And the number of cells at this point is going to be low. There are very nice new biosensors that tend to work really well in pure culture, in clean water. But then you're going to have some issues with your sample or an enrichment, or what is called like the matrix. What's in the matrix, like the turbidity, all the suspended solids, all that is going to affect this test kit. If you don't have a cleanup step before you test it, the machine has to deal with all these issues that they were meant to deal with. So they're meant to have it in a clean sample mainly. All right. So zero tolerance for pathogens presence. Zero tolerance, how do you define zero? What is zero? Well, zero is not really zero. It is how you define zero, and it's based on the sample scheme and the sample mass. So for pathogens, I was saying sample size 25 grams. So the contamination has to be heavy enough that if I grab 25 grams, X amount of samples, I have at least one cell in these 25 grams. But it's only so much you can test, right? Now it's tied also to the specific pretest methods and detection platforms that you will pick. Now the probability of detection, this is a qualitative test, and understanding the probability of detection under different circumstances and which one have been tested against the matrix and condition that you have is very important. Like I was saying before in the previous slide, you are going to have different, depends on the commodity, depends on how you process your sample. You're going to have, the matrix is going to vary by commodity. So make sure that, well, not only the test is meant to be used for that commodity under those circumstances, but also that has been performance tested. All right, so now understanding the limit of detection. So on the left, you have this graph where you are quantifying E. coli and E. coli 157 in soil that was originally in spinach and then was disked down, now it's in the soil. So we follow the survival of E. coli and attenuated 157 in the soil, and they survive in countable numbers based on our limit of detection, based on the sample size, for up to 110 days. After that, every time we wanted to quantify it, we couldn't, we had 0 counts. So we went from a quantitative analysis to a qualitative analysis in which you enrich your sample and then you grow whatever you have there. If you have at least one cell, that cell will duplicate and grow in your enrichment so you can detect it with the other techniques. But a good way to compare the limit of detection, imagine the horizon. You can only see as far as the horizon. You cannot see beyond the horizon. So you can quantify the location of the sun in the sky all the way until sunset. Beyond that, you know it's there, but you can't see it. So in an enrichment phase, it will be like increasing your, elevate your eye level so you can actually see what's actually there. Here, an example of with cantaloupes inoculated in the field. We enrich this cantaloupe. We know all these cantaloupes are contaminated with salmonella. At 0 hours of enrichment, we have 16 samples of all of them. We know all of them are positive, but only 16% were positive. By 10 hours, which is the incubation time that I was saying before for these rapid methods, we only had 83% of the samples positive. Now, like I said earlier in the very first slide, or the second slide, the FDA, people say usually why the FDA has more positive than we do. Well, you can take forever with your sample because you have a perishable product. So the FDA, on the other hand, they already know you have an issue, and so they have more time to incubate these enrichments. So you're increasing your chances of finding it. Finished product testing, I'm gonna go right through it. Safety cannot be tested into the product, and you have to decide what you're gonna test for. Negative test, don't prove absence. It tells you that there isn't some gross contamination event. If you have to decide, you're gonna test for the pathogen in concern, the target microorganism in concern. Focus on the process and not the product. Can resources be better used? Product testing is a decision to be made between the buyers and the suppliers. And I have the last 3 slides, probability of detection. In the past, I don't think this is something very common, but in the past, people will have a positive out of 2, for example, like in the first row, and they say, well, maybe that was a false positive, let's retest. And then when you retest, you're not increasing your chances of getting it again positive, you're actually decreasing your chances. So if you do a retest, you're going from 1 in 2, like you had originally, to a 1 in 4 chance. So if you have 1 in 10 samples, your chances of getting it again positive are 100, 1 in 100. So you can't say that because you test it again and it's negative, then the first sample was a false positive. Just that approach, you can't just have it. And then test kits that promote this 30-minute detection time, that seems too good to be true. Keep in mind that the total time to get to that before this 30-minute, it takes like about 28 to 30 hours at best. All right, so yes, the test itself at the end is 30 minutes, but you have to go through all these steps to get there. And degradation, there are many other things that I had to leave out. I just wanted, this is I think one of my last slides, degradation of dead cell DNA. There are many studies that on dead cell DNA persisting through disinfectants, through extreme pH, ethanol, UV, heat. And if reasonable to expect that it could occur on plants, then it will occur on testing treated process water. All right. So in this example, in this paper, I forgot to reference it, but listeria, dead cell DNA for listeria was detected up to 30 days, and for salmonella up to 55 days. All right, key take-home messages. Testing could be used to test around a bad practice or observation of concern. Microbial testing has a role in verifying your food safety performance. should be done only through protocol and only if a thorough protocol has been developed for that purpose, knowing what you are going to do when you get a positive result. And frequent negative results lead to give you a false sense of security. And know your sources of false negative and false positives. Do some investigative testing. If you have cumulative positives and you have some idea that something else might be triggering, invest that money, invest that time in finding out who these are, and see if you can get rid of that problem. These are some of the resources I left in the PDF, and I'll add on more of these in the actual files on Box, so you guys can download it, and you have to do it yourself. All right. So if you guys have any questions, I'll be here all day.